Hello and welcome to part 6 of chapter 6. Here we're going to talk about some anaerobic processes. In particular, we're going to talk about fermentation. So fermentation is an anaerobic process, but it utilizes glycolysis. And this anaerobic process, or the fermentation, occurs in two different forms. We have alcohol fermentation and lactic acid fermentation. Now, prokaryotic organisms such as bacteria most often take part in acid fermentation, and eukaryotic organisms such as yeasts will take part in alcohol fermentation. Fermentation actually occurs to pyruvate, not glucose directly. So glycolysis is the first step, and then fermentation is actually the second step. The purpose of fermentation is to recycle the NADH back into NAD+, so that ATP production will occur in glycolysis itself. So let's look real quick at glycolysis. In glycolysis, we always have to make some NADH right here before the ATP is made. So NAD plus is necessary, is required in this step right here, step 6, in order for step 7 and step 10 to actually occur. So we can't make any ATP in glycolysis unless we make NADH first. In order to make NADH, we must have NAD plus available. So in glycolysis, if glycolysis just continues, but there's no oxygen present, then acetyl-CoA formation and the Krebs cycle are not going to run. Those are not going to function, and pyruvate will begin to build up in the cell because it's not being further oxidized. Not only will pyruvate accumulate, but the NAD plus will continue to get used, but since there's no oxygen, there's no electron transport chain. And if there's no electron transport chain, then NADH will not be oxidized. If we don't oxidize NADH, we will eventually run out of NAD plus. No NAD plus, no NADH being produced, no more ATP. So fermentation is a recycling plant. It's a way to recycle the NADH back into NAD plus so that ATP production via glycolysis can occur. So here we have our two forms of fermentation. Here pyruvate is being converted into lactic acid or lactate. And again, you can see NADH comes in here. It gets oxidized in order to produce NAD plus. So the purpose of fermentation is the oxidation of NADH. In eukaryotic organisms with alcohol fermentation or ethanol, in ethanol fermentation, our pyruvate gets converted into ethanol via acetaldehyde. So this is actually a two-step process. You see we have two enzymes involved, whereas here in lactic acid fermentation we have one enzyme involved. By the way, our muscle cells, when they run out of oxygen, will carry out lactic acid fermentation, and that's where the lactic acid from exercising comes from. That makes you really sore. Now, catabolism of other molecules also occurs, and this is an important concept because we don't just break down glucose. It's not the only molecule that we use as a nutrient source. We use others, polysaccharides and disaccharides. We use other sugars like lactose, maltose, sucrose, table sugar, starch. So we do use other carbohydrates. We also break down lipids and proteins. You may have heard the expression that fats contain more energy than sugars do. And that's one of the reasons our bodies store energy in the form of fat, because we can get so much more energy from a fat molecule than we can from a single glucose. But these molecules have to go through their own initial pathways, and once they move through their own catabolic pathways, the products of their pathways then enter into central metabolism. For example, let's take a look at lipids. Lipids are glycerol molecules. I'm just going to put a GLYC here for a glycerol molecule, and attached to that glycerol molecule is a whole bunch of hydrocarbon chains. Okay, so we're going to look at a triglyceride here. So I have all these hydrocarbons. Remember, these guys have all these CHs on them. Now, when I break down this molecule in lipolysis, the first thing a cell will do is break off the will break off the glycerol molecule. So this glycerol molecule leaves, and if you follow it here, right, here's glycerol, you follow this arrow, glycerol will be converted into a G3P so that it can enter into glycolysis at the same place that the pentose phosphate pathway does. And so that G3P molecule now moves into glycolysis for ATP production in pyruvate. Now that leaves behind these hydrocarbon tails, and these hydrocarbon tails will be broken down in a process known as beta oxidation into little 2-carbon units. And so all of these little 2-carbon units, I'll put another one here just so we have it, all of these 2-carbon units, if we now follow the fatty acid line, will enter at the transition step and match up with acetyl-CoA. Now when this happens, if this were glucose, right, if I were doing just glucose here, I would get 2 pyruvates, which would give me 2 acetyl-CoAs. But because I am using a lipid, I get 1, 2, 3, 4. If this is times a triglyceride, 5, 6, 7, 8, 9, 10, 11, and 12, I would end up with 12 acetyl-CoAs from a single lipid molecule, whereas in glucose I only got 2. So I get more energy, right? I get more energy from a lipid molecule than I do from a glucose molecule, simply because I have all these hydrocarbon chains, and that lipids will be broken down and enter into the primary metabolism or central metabolism at the transition step. It skips glycolysis. Now we do have some exceptions to glucose breakdown, and those are a class of prokaryotic cells known as chemolithotrophs. Chemolithotrophs are organisms that will break down molecules other than glucose. So hydrogen bacteria break down hydrogen-based compounds in order to oxidize them, and their final product is hydrogen gas instead of water. Sulfur-loving bacteria will break down sulfur instead of glucose, and then iron bacteria and nitrifying bacteria. So these are the 4 different types of chemolithotrophs that use alternative forms of metabolism aside from primary or central metabolism. They extract their electrons from inorganic carbon sources as opposed to organic carbon sources. So chemoheterotrophs are the organic carbon using organisms. Chemolithotrophs are the inorganic carbon using organisms. And again, these are the 4 types: hydrogen, sulfur, iron, and nitrogen. So just a real quick review on this one. Make sure that you understand the process of fermentation, how it occurs in a cell. It's with or without oxygen. Why is it important in anaerobic conditions? Where do we get ATP from? These are the types of questions regarding both chemolithotrophs, or the 4 types of chemolithotrophs, and the difference between a chemolithotroph and chemoheterotroph, who is using what, and how ATP production occurs in fermentation. So this was a nice, quick, short little lecture. I will see you guys in part 6.